Good evening. Can you all hear me? Yes, very good. It's a great pleasure to be here today. Thank you, Lisa, for the lovely introduction. It is always a pleasure to present my work and my ideas, but it's extra special to have the opportunity to present to the broader community, not to a scientific audience, as I usually do. And so it's really a tremendous honor to be here with you today. So just the first thing that I wanted to say, as is important for all of us scientists who also have activities working with the biotech in the area, these are my disclosures. And then going back to my title, as I was preparing to my slides to give this presentation today, I was thinking that actually perhaps the most meaningful form of communicating one's scientific ideas is really in this format, speaking to the broader community. And the reason for that is because you are the stakeholders important stakeholders for everything that happens in this building, really. You are the potential recipients of the therapies, the benefits that can come from the research that we do here, first and foremost, of course. You are also our partners in this process. As you will see today, there is a theme that will carry us through my talk where the partnership with patients is actually critical to the success of precision medicine and treatment of diseases as we move into the future. And finally, of course, you're important stakeholders because, let's face it, a lot of the research that we do in this building costs a lot of money, and it is a lot of federal funding that goes into this research, and it is very important as you, the stakeholders, to know what goes on. And so I think it is the most important, critical part of my mission to be able to, as a scientist and as a physician, communicate with a broader audience about what it is that we do, what it is that happens in this building. So from that perspective, again, I'm deeply honored to be here. It also creates for me the responsibility to make sure that I'm clearly understood as we have our discussions today. And I have taken some time to make sure that my slides are free of jargon and in a way that would be understandable to a broader audience. And I also vetted my slides with two of my harshest critics, my son Kai and my son Dimitri, who are in the background, in 4th grade and 7th grade, respectively. And so they vetted my slides and said, that they would be acceptable. However, if at any point something does not seem understandable, please, by all means, raise your hand and let's find out what it is that wasn't understandable so that we can discuss it and move forward. So with that in mind, I wanted to start with asking, just looking at the title for the talk today, how many of you, maybe we can have a raise of hands, got the connection between unicorns and rare diseases? That's pretty good. What about the connection between zebras and rare diseases? Oh, that's still pretty good. Maybe we have more of a medical audience or people who have some connection to medicine. I tried the title out with a couple of friends, and someone said, I get the unicorn part, but what's up with the zebra? So I had to actually think about this for a moment because I assumed, it's one of those things, having been in medicine for a long time, that this is well-known, sort of public knowledge. Turns out that this expression from which this word zebra comes from to describe rare diseases dates back to the 1940s, and this man, Dr. Theodore Woodward, who was a beloved physician scientist at the University of Maryland, and who would tell his medical interns, when you hear hoofbeats, think of horses, not zebras, trying to suggest to them that they should not be thinking about the rare thing that they read in the textbook, but rather, when they're treating the patients, to think about the most common diseases. Now, interestingly, we will be talking about zebras today, and we will challenge a little bit, or perhaps extend the view of Dr. Woodward as to how much attention we might be paying to zebras versus horses, and are we really all zebras? Maybe that will be a concept that we might visit toward the end of this talk. So that's the zebras part. So what are we going to talk about today? I wanted to start by basically putting out the 3 topics that I would like to discuss with you. The first one will be about zebras and unicorns. We will talk about certain numbers and definitions around rare diseases and what they mean and what they are. And I will explain the concept of unicorns in that setting. Then we will take a deeper dive into an area that is of great interest to me, which is rare kidney diseases, where I've done some work. And I will share with you some of the work in that area. And then finally, we're going to look into the future and think about what lies ahead, what has worked, what needs to be done, what we can do better as we move into the future to develop therapies for rare diseases. And this concept, as I said, of are we all zebras, and how might we harness that for our benefit moving forward. So rare diseases. There are approximately 7,000 rare diseases out there. It's really a pretty substantial number. And if you look at the definition of rare disease in the United States, and that varies a little bit in different parts of the world, but in the United States, it is defined as any disease where there are less than 200,000 patients that suffer from that disease. And so that seems like a small number in general. You know, this is a very big country, so a lot of diseases and a small number that defines each of those diseases. But when you take them all together, rare diseases are not actually so rare. 1 in 10 Americans has a rare disease. So that's actually a pretty substantial number. And the impetus to really think about rare diseases or to look at disease through the prism of rare disease might actually be significant. And so as this youngster is studying the brochure about rare diseases and the numbers, 1 in 10 Americans suffering from rare disease, he's also learning that rare diseases are characterized by a particularly difficult problem. Most of us, when we have a symptom, are used to going into the doctor's office and maybe after 1 or 2 visits, usually there's some level of diagnosis and some idea about what might we do next about that problem. In patients with rare diseases, that does not turn out to be true. So the diagnostic odyssey, as people have called it, can last up to 5 to 7 years. That means it takes 5 to 7 years to even get to a possible answer as to what is wrong, never mind trying to get to a treatment for that problem. So you can imagine that can be a really difficult, emotionally difficult journey, as well as one that can be very expensive, that can also cause the progression of a disease that has not been adequately diagnosed because there's no available treatment while there's no diagnosis. And so as you can imagine, the burden on patients with rare diseases, and often children and their families, as it would be, is quite significant. So this brings us to the subject of unicorns, which I wanted to discuss. Unicorns, since ancient times, and in this Renaissance painting, were thought to be sort of mythical, mysterious, elusive creatures. I mean, there's a lot of discussion. I went on the internet and saw that there are some people who claim they have seen a unicorn, but there's no real scientific evidence that a unicorn actually exists. And very much so like that, it is true for some of the diagnosis of rare diseases. They are what I would call unicorns, meaning they're elusive, they're mysterious. Maybe they're right, maybe they're wrong. Have we seen it? Do we know what it is? Maybe not. And it actually turns out that this is a real problem. About 50% of all patients with rare diseases end up with a unicorn, not a real diagnosis. They're not a real zebra. They're a unicorn that poses as a zebra. And that can be a real problem, because that basically means that there's this elusive understanding of what may be wrong with them, which actually prevents them from potentially moving forward with any possible definitive treatment, if that would exist, for their disease. So there are pretty significant barriers, and dealing with unicorns is one of the problems in this space. A very good example of the journey, of a diagnostic journey that has some answer, maybe not a happy ending. We will have some stories today that I will share with you, and not all of them will have a happy ending, but a story that is of great interest from the perspective of the diagnostic journey is the story of Bertrand. Bertrand is this young boy. Bertrand Might is his name. If you have not read this article in the New Yorker from back in 2014, it's really a wonderfully told story. Bertrand is the son of 2 highly educated professionals living in Utah. They had their first baby, and they named him after Bertrand Russell, the philosopher. They had high hopes for their baby, like everyone does. But they noticed pretty much soon after he was born that their baby was what they describe as floppy. There's a description in the article that says that it felt as though he was made of jello somehow. He couldn't keep himself propped up. And you can see that Bertrand is lying here on his bed. No one could figure out what was wrong with Bertrand. They went from one specialist to the next, and they did one test and then the next test. And no one really knew what the answer was, what was wrong with Bertrand. Soon enough, they started to realize as he was starting to grow up that he had some developmental delays. And again, tests were done. They went from one specialist to the next. These are people who are highly educated and able to tap into all kinds of resources. They searched high and low all over the country to look for specialists who might be able to give them the answer. And at the end, they had an answer. And that answer came from doing sequencing, looking at the genome of Bertrand and trying to find out if there was a mutation that might be causing his disease. And so that's one of the themes that I would like to bring out today. So Bertrand had his genome sequenced. That's, all of his genes in his genome were sequenced. And ultimately, and actually that happened in places like the Broad, where we can, do this kind of sequencing work. I will describe that further in a little bit. And ultimately, multiple scientists came together and found that the mutation that was likely to be causing Bertrand's problem was a mutation in an enzyme, which doesn't, the details don't really matter, but it's called NGLY1. And it turns out that at the time, as the article in the New Yorker said, Bertrand was the only human being on the entire planet that seemed to have this mutation. So that's why the article was entitled One of a Kind. Well, it turns out that his father, who's a professor of computer science and very well connected on the internet, was able to post a blog and tell Bertrand's story. And soon enough, there was sharing of the story. It was throughout the internet and reached faraway places like California and like India, where the second and third child with this mutation was ultimately found. And at this point, now we have a handful of people with this mutation. So now all of a sudden, this has become a new rare disease that has been defined through genetics, through the investigation of genetic information. And it is now called NGLY1 deficiency instead of Bertrand's disease. So this shows you the power of being able to bring people together, having incredible patient advocates. In this case, that's Matt Might, that's Bertrand's father, who has really taken patient advocacy to a whole new level, as is often the case with parents of children with rare diseases, has founded this organization which is now looking for treatments for Bertrand's disease, for NGLY1 deficiency, and who, as you can see, was recognized by NORD, the National Organization for Rare Disorders, which is one of the main patient advocacy groups for rare diseases. He was recognized this year with the Rare Impact Award. So I tell this story to give you an example of the diagnostic journey that many patients face, and to really bring out some themes about genomics, about patient advocacy, about parents looking high and low for an answer to their child's problem. And this is where Bertrand's story is, the arc of his story is at this time. They're still looking for a treatment. They know the cause of the disease, but now comes the phase of trying to find a treatment for the disease. But as I said, one of the main themes that I wanted to really discuss with you today, after all, we are in the Broad Institute, is the power of genomics. And this comes out again and again in many of these stories about patients, children and adults with rare diseases. Now, I don't know how much you may remember of this, depending on the different backgrounds that people may have in the audience. But as you know, we human beings are defined by 23 pairs of chromosomes. And actually, if you look at our genetic code, which is encoded by these combinations of these four letters, A, T, and C, and G, there are 3 billion such base pairs in our genomes, okay, that encode the genetic information that makes us who we are. And it is an incredible process because even one mistake, and I'm giving you an example here. Here's one mistake where C is replaced by T. And so instead of now, let's say, reading cat, now you're reading tat. Well, you can see how the process can go awry. And so the genome is now not producing the right thing. It's not producing cat. It's producing tat. And that's not what it's supposed to be. That's how mutations occur. There are many other mechanisms by which they occur. But it's just a simple example to illustrate that this is the kind of thing that can happen when these base pairs are somehow not connecting to one another as they should, and mutations occur, and that leads to abnormal molecules and diseases in children as well as adults. So the power of genomics lies in our ability to be able to detect these mutations. And I just want us all to remember that this was not always the case. We now live in a time when we can do this, but this was not always the case. So I want to remind you that it took 20 years for one human genome to be sequenced, and this happened to a great extent here at the Broad, actually, but a lot of other places around the world. It was a massive group effort by a lot of scientists. And now, at the Broad, we can sequence a whole human genome in about 10 minutes. If you just think about the scale of these events, it's just enormous. And it really allows us to now have the ability to read thousands of human genomes and start to make conclusions about what may be the cause of disease, the drivers of disease, and how we can target that for therapy. And so the fundamental premise now for how we might be able to get there is the sharing of those thousands of genomes. If I, as a scientist, would sequence 1,000 people, and you, as a scientist, would sequence another 1,000 people, and we kept it to ourselves, we would never be able to get anywhere. This is a numbers game. We need to bring all of the data together so that we can all start to decipher, which are the real mutations that cause disease versus, noise in the system, as one would say. And so the Broad has taken a lot of leadership in this area as well. This is through the efforts of a very talented colleague of ours, Daniel MacArthur, and I just wanted to point out that he has been instrumental with countless other scientists here at Broad to create this genome aggregation database, which is essentially that. It contains at this point 123,000 exomes. These are, the exomes are sequencing of the parts of our genome that encode for specific proteins. There's a kind of 99% of the human genome that does not encode for any known proteins. And that's looking at the entire genome. So we have about 15,000 genomes right now and about 123 exomes. So exomes are kind of an easier, faster, shorter way of getting to the genetic information. But genomes contain more complete information about our human genomes. That's one way to think about it. So that's where the database stands right now. And I bet that it will increase ever so over the next few years. And it will become a tremendous resource, as it already is, for a lot of this discovery, including fueling our ability to discover mutations that cause rare diseases. So I just want to make sure that this point is clear. When you're sequencing someone's genome, you find a lot of mistakes. But how do you know which one of those mistakes is the one that drives to disease? There may be thousands of such little mistakes there. So the way you do it is by comparing to thousands and thousands of other genomes and finding out which of these mistakes might be unique to this individual versus common to countless other individuals. And by going through this exercise and using pretty sophisticated math, you can start to decipher which of these mistakes, which of these mutations might be real and might be driving disease. So it is absolutely essential that we go through this process of aggregating in a database all of these genomic data and that we all contribute to that so that we can be able to decipher mutations, which ones are real and which ones are driving disease, so we can help those with rare diseases. So it's absolutely instrumental. And of course, we are not going to stay idle. We at the Broad are always moving forward and always wanting to do the next big thing. And so the next big thing, which is also spearheaded by Daniel MacArthur's group here, and with whom I have the great honor to collaborate on some of this, is the Rare Genomes Project. So rather than just going and aggregating as many genomes as you can get out there from all kinds of different patients with all kinds of different diseases, there's also the possibility for making a focused effort on aggregating genomes from patients with rare diseases and trying to compare them to those other genomes that have no rare disease and trying to see what you might be able to find. So the Rare Genomes Project is doing something very unique, something that I think will be it will change the paradigm for how scientists, genomic scientists interact with patients. And that is, we would go directly to patients. We actually have already started this, and Haley Brooks was very important in putting all of this together, working with Daniel, is I think here tonight. And so the Rare Genomes Project is really an amazing effort to try to say, it's what someone said to me the other day, is the complete democratization of this process. Basically, the way that I would describe it is, if you have internet, a computer, and a mailbox, you can participate in this study no matter where you live. You could be living in the middle of farmland someplace, I don't know, in Montana, and you can still participate in our study as far away as you might be from an academic medical center. And the idea is, that people can go to this website, they can answer a short questionnaire, they can then be enrolled in the study. We would send them a kit, and they can send us back their blood, their saliva, whatever material we might need in order to have them participate in a study, in order for their genomes to be sequenced free of charge to the patient, so that we can actually start to assemble this essential genomic information for deciphering what the answer might be to these rare diseases. So this is a project that is just getting started. We are very much looking for partnership from patients everywhere. It is instrumental that patients participate in order for this to be a success. And so we're very much looking forward to trying out this direct-to-patient approach and seeing how people out there see this. There's already been tremendous enthusiasm for this effort. But of course, genomics will get us to diagnosis. And yet, when you talk to these patients and their families, what they're really after, of course, is treatment, right? They are sick and they want treatment. And of course, it is absolutely essential that we make a diagnosis before we can provide a treatment. That's one of the main things that we teach our medical students, of course, day in and day out in the hospital. But treatment is what these families are after. And this is just beautiful portraits. I don't know if you recognize this, but this is Bertrand, the boy who we met earlier. This is a tremendous idea from the Rare Diseases United Foundation. They have organized an exhibit, which we have actually hosted twice now here at the Broad. These are portraits of kids with rare diseases, and they were painted and donated by various painters around the world. There are now hundreds of such portraits that they have collected, and it's a vehicle with which they can raise awareness. And as one of the people in charge of the foundation was telling me, they have raised more awareness by bringing this exhibit to different parts around the country and actually around the world now too, than by any brochure, any website, or any other means with which they have tried to reach people. It seems as though the pictures are really able to capture people's attention and really raise awareness for rare disease in a way that was not previously possible. So these kids are telling us, we need a diagnosis, and now we also need a therapy. And it is that concept of working on therapies that really attracted me to this space. I very much am interested in developing therapies for people with rare diseases, and that is what I have devoted my career so far to doing. And that brings us into the particular area of rare disease that I, at the moment, am focusing on, and that is the area of rare kidney diseases. So, as with everything, that also starts with a story that starts many years ago and in a place far away from here. This is the city of Thessaloniki in Greece, which happens to be my birthplace. So I was born and raised in Thessaloniki, and I happened to also be the daughter of a kidney specialist. And my dad would often come home and tell me stories about what happened in the hospital. And one day he told me about the story of George. And George and I were about the same age when my dad was telling me this story, and it really sort of captured me. George had a rare kidney disease called nephrotic syndrome. I had never heard of that before. He had been sick since he was very young. There was no cure for his disease. And George's kidneys had failed. And so he was actually on dialysis, which is a machine that you get hooked up to clean your blood out a few times a week. It's a way to replace the kidney function. We will hear more about it in a little bit. And he was, of course, waiting for a kidney transplant. I was really taken by this story. It was a kid who was about my age. George was basically tethered to the dialysis machine a few times a week and could not really attend summer camp, for example, or any of the other activities that most of us were looking forward to. And of course, I felt really bad for George. And I was right to do so because waiting for a transplant is really no small task. As you can see here, this is data from the United States, but we have a real supply and demand problem here. As you can see over the years, the demand for kidneys, essentially, has increased, but the supply has remained pretty steady. There's not a lot of kidneys available to people who are waiting for a transplant. And in fact, it is true that many people die waiting for a kidney transplant. And so that's a real problem. We don't have a good solution. But here I was 14, 15 years old, and didn't really know what to do to help George. But I came up with an idea. This is a picture of my high school. I attended an American school in Thessaloniki. It's called Anatolia. It was founded in 1886 by American missionaries, and this is what it looks like today. So I couldn't help George in any sort of practical way. So I decided that I would sign up for an oratory competition, and I would give a speech about George's story and about transplantation and try to urge people to become kidney donors. And so I ended my speech with this phrase: ladies and gentlemen, don't take your organs to heaven. Heaven knows we need them here. And it seemed that the talk was pretty impactful. A lot of people came up afterwards and said that it really got them thinking. Maybe I was able to convince some people to sign up to become organ donors down the road, who knows. the arc of George's story at this point bends toward happiness in the sense that he did actually end up getting a kidney and having a kidney transplant, and that was, of course, a wonderful thing. But this experience also taught me about the power of words and communication, and this is what we're doing here today as well, the idea of being able to communicate a message that can then resonate with people and allow them to think about their own lives and how they might take this information and how they might change certain ways of thinking about their activities or their ideas down the road. And so that was another powerful lesson that I learned from this story. Okay, now we fast forward many years. I was incredibly lucky to get a scholarship to come to Boston for college. And past that, I was working on a PhD. And of course, I was kind of the feisty daughter of a kidney specialist who would never become a kidney specialist. And so I was going to become a brain scientist, a neuroscientist. And so I was hard at work in the lab working on neurons, on brain cells. And the details really in this graphic don't matter too much, but I was essentially for the better part of 7 years focused on essentially these two molecules, TRPC5 and RAC1. And to make a long story short, during brain development in certain parts of the brain, I discovered that these little finger-like projections of these brain cells move to kind of sniff their way around and find a target, another brain cell with which they form a connection. That's called a synapse. And that's how brain cells talk to one another. That's how our brains fundamentally work, by cells connecting to one another, brain cells connecting to one another. And I thought, okay, I mean, this is interesting. These molecules are clearly important in the brain, and probably I will become a brain specialist down the road, and that's how things are gonna go. But fate had different plans for me, it would seem. And so when I went back, I finished my PhD and went back to do further medical training. At some point, I came across these papers that came out, and they basically, I was reading, and they said, oh, here's RAC1. Okay, I know this molecule. I encountered it in my PhD. Oh, but it's actually important in these cells called podocytes, which I hadn't really known as much about. And there are mutations in this molecule. And I thought, oh my God, and they don't cause any brain problems as it would be. And people have checked, these patients do not have any brain problems. But they have this nephrotic syndrome, which that's George's disease, And so it sort of started to resonate that, wow, these molecules that I obsessed over, truly, for many years during my PhD, were now becoming relevant to nephrotic syndrome. So I was sort of coming back to all of that. So that was sort of one of those moments. The light bulb went off, and I said, okay, maybe I really should think about nephrotic syndrome. Maybe I really should think about kidney disease. Maybe I'm meant to think about kidney disease. And so I went back and started to think about nephrotic syndrome again. And unfortunately, since the time that I was a teenager, not much had changed. Actually, to this day where I stand before you, barely anything has changed, and that's what I'm trying to do to make a positive change with that. So nephrotic syndrome is a rare kidney disease without a cure. It affects 1 out of 2,000 people, just about. It affects children and adults. And as you can see, this little boy here, and we'll see a little bit more about the mechanisms of how this disease occurs, but it results in people swelling up, and oftentimes patients will say that they woke up with their eyes swollen shut, and that was one of the first ways they knew that they had the disease. And of course, that's a pretty awful thing to happen, and so brings them to the doctor, and then ultimately the diagnosis is made. And so I decided that I should start to think a little bit more about this. So what happens to someone with a kidney disease, a rare kidney disease that has no cure? We talked a little bit about George. There are 2 options. There's dialysis, which is, again, connecting with a blood filtering machine about 3 times a week. Unfortunately, if you actually look at the statistics for dialysis, they're pretty grim. So within 5 years, 7 out of 10 patients no longer survive. These kinds of statistics are worse than most cancers these days, because we've gotten pretty good about treating a lot of cancer out there. And so these are pretty grim statistics for kidney disease. And kidney transplant, of course, offers a hope, but actually it is a little bit sombering to think that 6 out of 10 patients will not have a kidney that works after 10 years. And that is where George's stories, the arc of George's story bends in a negative direction. He actually did end up having a kidney that stopped working after he got his transplant. And he actually passed away at the age of 22. from complications. And so that also tells you that these things are not an answer. You know, they are a stopgap and they may prolong life for a while, but the quality of life is not very good and it's not very long. And so we should do better. We should do better for these patients. So I felt like this is something that I really wanted to devote myself to. And so that would mean that I would really have to now turn myself from a brain scientist into a kidney scientist and really try to understand what's going on. And in a simple way for me to explain what goes on in these kidneys is if you would look inside a healthy kidney, you know, the kidney, one of its main roles is to filter blood. So if you can imagine two compartments, there is the top compartment that has the blood in it, and then there's the bottom compartment where the urine starts to form. And in between them is a filter. And normally, proteins, like albumin, which is the most abundant protein that we have in our bloodstreams, would stay behind. This filter would be there to take away extra electrolytes or toxins, but it's not supposed to let proteins go through. That's not how nature designed this exquisite filtering system. However, inside a damaged kidney, this is what happens. This filter breaks down. That's what happens in nephrotic syndrome. And these proteins, albumin, that were not supposed to spill into the urine, now spill into the urine. This is called proteinuria, and this is the hallmark of nephrotic syndrome for all of these patients. Furthermore, I delved further into this to try to understand a little bit more. And what you can see if you would blow up this filter that I was just describing is that it's composed of basically two layers of cells. These other cells up here, which we will not talk about today, and the cells that I would like to focus on, which are called podocytes. These are the cells that are sort of on the urine side of the filter, if you like. And it turns out that they are pretty important. They are the ones that, when damaged, really lead to a lot of trouble. So nephrotic syndrome is the result of these precious podocytes getting injured and dying. And they're precious because we're all born with a certain number of podocytes, and we, to this day, have not figured out a way that these cells are replenished. So, you know, you're born with a certain number, and if you start losing them, that's it. There's no way to replace these cells. Every other cell in the kidney can be replaced except for these cells. And so they're pretty precious. And it turns out that when you lose them is when you get into trouble. So that's what was known about this disease. And as I mentioned, when I was in medical training, I read that paper about the RAC mutations, right? So again, you know, this is a mutation, a misspelling in the base pairing in the DNA. And so in this case, this mutation was causing the podocytes to get injured and to fall out. And it seemed that that was caused by this mutation in this molecule called RAC1, causing too much RAC1 to be around. Well, that was interesting to me because remember, when I was studying the brain, you know, I was interested in the connection between RAC1 and TRPC5, this other molecule. And what came to be understood, and by now I had my own lab and I was sort of studying this disease in my own laboratory as a faculty member at Harvard Medical School. What was clear was that once you have a mutation that causes more RAC1 to be around, what you end up with is more TRPC5. And that is not good for the cells. That's how podocytes die. So of course, we're scientists and we form hypotheses. And so that resulted in us forming the hypothesis, what if we were to shut down TRPC5? Could we rescue podocytes? And could we rescue kidneys? Right? That seems like a logical hypothesis to make. And so we went about testing that hypothesis in the laboratory. And so now you're poised to meet your first podocyte, perhaps. This is a podocyte that has been injured. And as you can see, the things that make it stand up, get propped up, it's called the cytoskeleton of the cell, under the stress of injury, collapses. The cell shrivels up, essentially, and ultimately dies. But here are some podocytes that were injured, but also had the opportunity to receive a drug that blocks TRPC5. And when you look at them, you can see that they do not shrivel up. They stay propped up, and they seem pretty happy. So we were pretty excited about that because it seemed as though shutting down TRPC5 might actually work in cells. That's where everybody starts to look first. You know, we scientists, you know, start small, and then we start to go bigger. And bigger means testing the same idea in mice. And so here's how that experiment goes. Whoops. Here we have two kinds of mice. a mouse that is normal and a mouse that has damaged kidneys. And these mice get nothing. And these mice get placebo, actually. And these mice get an injection with that drug that we just saw was keeping the podocytes propped up and healthy. And when we looked at these damaged mice, the mice with the red kidneys, so to speak, we saw that the ones that received the drug had far less damage, and that is measured as the albumin, the protein that was spilling into their urine. Remember that graphic that we saw earlier? So it seemed as though the mice were getting protected from the injury after they received this blocker of TRPC5. So that was pretty exciting. This is work that happened a few years ago in the laboratory, and we have since moved on to pursuing more and more work in this area. And we hope that this might have show some promise to become a therapy down the road for patients with nephrotic syndrome, at least patients who may have overactive RAC, you know, the connection between the RAC1 and the TRPC5 that I mentioned. However, going from genetic mutation to hypothesis to confirming a couple of hypotheses in cells and in mice, all the way to developing a therapy into humans, it's probably going to be another 10 years, 15 years before we can get there to actually make a drug, assuming that it works, that we can put into a human being. That's a very long road. And our patients need an answer. We should be working harder than that. And we are working harder than that. And so there's another idea for rare diseases that I really wanted to talk about today, and that is drug repurposing. So this kaleidoscope, the details of which do not matter too much, is meant to show you that at the Broad, we have what we think is perhaps one of the most comprehensive repurposing drug libraries in the world at the moment. It contains 4,500 treatments or drugs that have already been developed. They are already out there for another disease. And so anytime you're dealing with a rare disease, if you understand something about its mechanism, or if you have a way that you can check if a drug would benefit your cells or not, as I showed you before with the podocytes, you are always able, here at the Broad, to go and check against this library of compounds. Is there one out there that I could just reach for? A drug that is already approved by the FDA, a drug that another patient is taking for another disease, a safe drug that you can now use to treat this disease, this rare disease right here. Turns out that this drug repurposing is actually a pretty good idea for rare diseases, and it can really shorten the time to treatment. So we can harness the investment that we all collectively have already made into developing these drugs for other diseases and design smart ways to find out whether they might work for our disease of interest here. And so again, the Broad is very well poised to do this kind of work moving into the future. This is a library that has been assembled over the last few years. Todd Golub and Steve Corsello have been a big part of this effort, but again, many, many other scientists. You know, at the Broad, science is a team sport, and it takes really a lot of scientists to make this work. So we have assembled this library, which we can use as a way to look for treatments for rare diseases. And in fact, we are very much engaged in doing this kind of work here at the Broad for a number of rare diseases that our team is working on. So repurposing is a story that I wanted to tell you next, and that's the story of Mary. So Mary's rare disease is also nephrotic syndrome, a rare disease otherwise known as focal segmental glomerulosclerosis. It's a mouthful, never mind. It's nephrotic syndrome. This is the best picture that Mary could give me. She has given me permission to show her picture. She's one of my patients. And she doesn't have a better picture from the time that she was sick. She doesn't want to have a better picture from the time that she was sick because she really didn't feel very well and didn't feel like having her picture taken. She was diagnosed when she was 9 years old and did not really respond to any of the treatments that were out there, many of which are pretty toxic and nonspecific, and they caused her a lot of problems growing up. So she was in my clinic. I had just met her. She was a new patient to me. She had advanced from pediatrics into adult kidney doctors. And I was meeting her for the first time. And we were sort of looking at the fact that there really wasn't an available treatment for her. And here she was with full-blown disease and no answer. Now, the Munda laboratory down the hall from mine had discovered that a drug called abatacept, which had been approved by the FDA for another disease called rheumatoid arthritis, might work to rescue podocytes. They knew that in the Petri dish. And so, of course, the question that I asked was, could that work for Mary, right? Could we repurpose this drug? Now, this is a drug that has been approved for a number of years, both for rheumatoid arthritis as well as for juvenile rheumatoid arthritis, which means kids with rheumatoid arthritis. It has a pretty good track record, and it's definitely safe as far as approval from the FDA. It has already been approved. It has a track record. People know about how to use this drug. So it felt reasonable to try it in a different disease, but it is also a bit of a leap. So I partnered with Mary, and we sat together, and I said, you're going to be the first human being ever to receive this drug for this disease. Countless others have received it for other reasons, but no one has ever received it for this disease. I can pretty confidently say that it's going to be safe. I cannot tell you if it's going to work. What do you think? This is one of those sort of valuable moments of the doctor-to-patient relationship and that trust that develops between people. And Mary said, I want to give this a try. And so we did. And so this is what happened. I think the before and after picture speaks for itself. Mary was an amazing responder to the drug. We got very lucky, I would think, now in retrospect. We gave her the drug, and first she had going in the wrong direction. Anything above this black line is bad in terms of spilling of proteins into the urine, and anything below this line is good. So finally, we got into the good direction, and she was doing very well. And this was amazing for her because this was the only treatment that she was receiving. She wasn't taking any other pills, any other medicines, and this was working really well for her. And Mary was able to, she finished college and was able to go to full-time work and sort of have her life back and feel like a real human being. And this was just transformational for her. Mary has been on this treatment now for a number of years. We're almost closing into 5 years. And she's what I would call an exceptional responder. Not everybody out there has responded to this drug in the same way. There are others who have responded similarly to Mary, and there are many who have not responded at all. There's something about this that we need to understand further. And we are doing this by trying to understand the genomics of Mary. Mary is our exceptional responder. We will also be looking for other exceptional responders and comparing their genomes to those who are non-responders to try to decipher this. I don't have the answer for you to that story yet, but we're working on it. But this is why she's an exceptional responder, because every time we've taken the drug away, you see these little gaps. Mary shot up, but then every time you put the drug back on, Mary responds. So now, of course, we've learned our lesson, and we're not going to be taking the drug off again. She's very comfortable staying on the drug for thank you very much, and she's doing very, very well. In fact, I just spoke to her a couple of days ago, and I said, I'm showing your picture. Are you okay with that? And she said, by all means. She wants to be an advocate and sort of have you all think about how important it was for her to have this repurposing happen. Yes?
Yes, so sorry to interrupt.
No problem.
While this is on the screen here, we see that the curve going up with the treatment.
Yes.
And what makes you, because it looks like as if it gets worse, so what makes you as a physician think to keep on going? Because
right.
It looks like as if
that's a good point, yeah. So abatacept is a biologic medicine. It is not a small molecule, and it takes some time to load it into a patient. So this bump here occurred very soon after she got her first or second dose. And then she got a cold, which we knew with Mary was a trigger for her to get sicker. And so in the face of that, we sort of kept the medicine at bay for a little while. We let her recover from her cold, but then we started over again. Because when I first gave it to her, as you can see, she was here when I first started, and she dipped down. That was a good sign. But then she went up again, and I was like, oh, that was a moment of reckoning in some ways. But we decided that we wanted to sort of continue to give it a fair shot, because a lot of these biologic medicines need some time before they load and before you can actually see their full effect. And so as a result, we persisted. I'm not showing you here all the doses as they were given. I'm just showing it as a flat sort of bar. But they were actually distinct doses that were given. And so once she had 4 doses, she was below the line and safely below the line for a long time after that. So that's what kind of helped us get going. Now, you're also touching on something else, which is the mechanism for how this drug works is also to suppress the immune system. And so it is possible that she was more prone to that cold that ultimately, kind of shot us in the foot initially. But we agreed to persist, and actually, since that time, she has not had, when she gets a cold, which she does, it does not trigger this kind of thing anymore, which was always the case since she was 9 years old. And so that's actually kind of remarkable that once you got the medicine in her system and it stays there, it's able to suppress her from getting into further trouble. At least that's the interpretation that we have at the moment. So that's the best that we know about this therapy. But it is a successful attempt at repurposing, something that you could immediately pull off the shelf and give it to a patient with a very strong rationale, meaning I didn't show you that data, but the Munda lab had produced amazing data to show that this drug really worked in cells and in animals before we gave it to Mary. But again, it was a safe drug that you could just pull off the shelf and repurpose. So it's a strategy that can work well for a lot of patients with rare diseases. Yes?
When you repurpose, though, I mean, the FDA approves drugs for a purpose. When you repurpose, do you have to...
Yes, so that gets me to the next point, which is, yes, when you're doing it as a one thing, as a physician, you can do an off-label use, of course, for a drug if you think that might work for a patient or two. But if you really want to kind of change the label, so to speak, and really have this disease be included in the indications for the drug, what you have to do is a clinical trial. And so these are my building blocks for what I call the one to more to hopefully all, or at least to many, as I show here. So Mary is the one. We ended up treating 5 patients total, and that was the story in the New England Journal. And people said, oh, podocyte-targeted therapy, maybe there's something to that. I still think that that's true. We just need to do more in that regard. But in terms of this particular story, if we really want to understand exactly how this drug works, we have to do a clinical trial, and that's exactly what we're doing right now. We're enrolling patients in 20 centers all across the United States, and we're going to test the effect of this drug in different patients in the hopes of being able to identify who the responders are, and actually use genomics to find out why they were responders versus those who didn't respond, and really sort of try to crack the puzzle that way. So that's something that we're very eager to do. In this case, in this repurposing case, it's a little bit like we're reverse engineering things. Like in other rare diseases, we might start with the genomics and then get to the therapy. Here, the biology drove us to the therapy, and now we have to find the genomics that will help us find who the patients are, the precision medicine part of this story. So we're working hard at this. The study started last year, and we are enrolling patients, and we'll see what we find. But it's a long road. That's what we need to understand how these treatments work. So as we continue our discussions today, the final piece, which is the shorter piece of my talk, really speaks to looking into the future. What have we learned, and what can we do better as we move forward? So I didn't really speak to that, but the impetus for a lot of these treatments for rare diseases really comes from legislation that passed a long time ago now, in 1983, the Orphan Drug Act, which actually incentivized in some ways drug companies to invest in treatments for rare diseases. And there have been many drugs that have received a rare disease designation as a result of this legislation. And actually, even more importantly, perhaps, drugs that have actually been approved under this designation for use in patients with rare or orphan diseases, as they are called. So what comes next? Well, it's a little bit sombering to think that despite all of this effort and all these years, we only still have the treatment for 5% of all rare diseases. So we really have a lot of work to do. There are many drugs currently in the pipeline that have a rare disease designation or pining for that, and I think that's really good. We know that there's a pathway forward now, so we can pursue that. Genomics, I would argue, is at the core of how we pursue treatments for rare diseases moving forward. And I shared with you Bertrand's story, which I think really illustrates that. And I think the final piece that is really sort of my message to you today is that partnering with patients is going to be critical as we move forward. The relationship with patients as we move into the era of precision medicine is going to become ever more important as we continue to look at genomics and precision therapies for patients as the driving force. And this cycle is really what illustrates that essential partnership with patients, which I think everyone in the Kendall Square area who's working in the rare diseases space, including people in biotech, will recognize as being an important cycle for the development of new treatments. We start with patients, red patients and green patients, but there's patients of all different colors. We're all different in many different ways because our genomes are different. You can find disease-causing genetic mutations by doing the sequencing efforts that we mentioned earlier. Then what you have to do is identify molecules that can be easily detected, for example, in my blood, that can tell you whether I have the disease or I do not have the disease because the treatment has addressed the problem. So that is what we call biomarker work. It's actually exceedingly difficult to do, but it is absolutely essential if we're going to make precision medicines possible for patients. And ultimately, that leads to being able to match a green pill to a green patient, meaning the right pill at the right time for the right patient. That's what this cycle is all about. And you may need to iterate and iterate and iterate until you can get there. Now, it is true that we shouldn't be in it only because we want to treat rare diseases. And there are spectacular examples of people going after a rare disease and ending up with the most amazing therapy that can be given to millions of people out there. And this is one of the examples that I like to give. Michael Brown and Joe Goldstein were two physician scientists. They trained here in Boston, and they ultimately went to Texas. And before getting to Texas, where they assumed their professorships, they made a stop at their time, as they were growing up as physician scientists. It was a requisite stop at the NIH. They went there for training. And at the NIH, they met a young girl, a 6-year-old girl, who had familial hypercholesterolemia. This is a very rare disease that results in cholesterol buildup in the arteries of small little kids so that they get heart attacks when they're like 6 or 7 or 8 years old. It's really amazing. So they met this girl, and that led them to spectacular Nobel Prize winning work that ended up resulting in the identification of the LDL receptor and drugs which are officially known as HMG-CoA reductase inhibitors, but more commonly known as statins, which I bet a very good percentage of the people in this room are taking. So they were after finding an answer for this little girl. But what they developed is one of the most successful treatments ever to be discovered so far. And so this illustrates that focusing on rare disease, which means focusing on the cause, the molecule that is responsible for the problem, and the treatment based on that molecule, can be actually very fruitful for more common diseases as well. Again, this being a spectacular example. And so for my last slide, precision medicine is what we're always talking about. What is it? What does it mean? And maybe in the context of precision medicine, we're all zebras, meaning we're all unique. We all have unique features. And as we look for treatments, we might look to our genomes to try to find the right match. So to me, a way to summarize precision medicine is the right treatment for the right person at the right time. And we do need genomic information in order to make that possible. And from that perspective, we're all unique. Our genomic information is unique. And so we have to take that into account as we try to design treatments, as we try to make decisions about our health. Rare diseases, I would say, have been leading the way in this approach. And so the lessons that we learn from rare diseases, again, may apply to all kinds of diseases down the road. And importantly, in terms of advocating and organizing partnerships with patients, which I think are very important. Of course, the Broad Institute has been at the leading edge of all of the genomics work, and now the leading edge of the therapeutics work, and we're ideally poised for doing work in rare diseases, as well as more common diseases based on the paradigms that I just described. So I think that it's going to be exciting times ahead, and I'm very excited for you all to follow us as we take our discoveries to the next stage. So with that, I just wanted to thank the people who are responsible for a lot of the work that I presented today, and it really takes a village to get all this work done. I have the privilege to work with incredible scientists here at Broad and at the Brigham, Harvard Medical School. This is a picture of our team from a couple of years ago, just a small subset of our team, actually. And there are multiple other collaborators across the institution and actually across the United States and the world who make this work possible, of course. I just wanted to acknowledge patients. Patients have really taught me a lot throughout the years, and I think it's incredible to have the opportunity and the privilege to take care of them, as well as to partner with them in this voyage of discovery as we continue. So with that, I will thank you for your attention and take any other questions you may have.
So we have time for just 1 or 2 questions before the reception.
Lisa? Sorry.
Do you coordinate with, since the size of the databases are really important, do you coordinate with, say, 23andMe out of California, which has my DNA?
Mm-hmm. Yeah, that's a...
10 years at least.
That's a good question, yeah. I think that there's an opportunity to... commercial entities need to make their data openly available as well. I think this is an ongoing discussion about how this is going to happen as we move forward. The Broad has really clearly put a stake in the ground about the fact that data should be open. And we hope that everybody will agree. I think it's becoming more and more apparent that this is the only way forward. And I do think that that's sort of the wave of the future. So. Yeah, there will increasingly be ways to volunteer your data through a website so that you can be what we call counted in. So the opportunity to be counted in for genomic analysis and making that data available to the world so that everyone can benefit from that information. So yes, that will be something that will be available. Yes. Oh, sorry. The gentleman there is waiting too, but yeah. Yes.
One of the science of this and the partnership and your beautifully lucid talk seem like it could make things move very, very quickly. But in the United States right now, we have had the GINA law, which has no teeth in it, that is supposed to protect people's genetic information from leading to not being hired or being fired. It has never involved disability insurance or life insurance, which is actually required to buy a mortgage in many places. And about 6 weeks ago, a law was passed that effectively said employers could force you to have genetic tests and reveal the information to them. So in terms of the larger scale organization and advocacy that has to be done, I think that at this point, that's the rate limiting factor.
I think you bring some very important points forward, and I couldn't profess to have the answers to these problems, but they are exactly the problems that we are grappling with right now. I think it's going to take advocacy, as you say. We are in a very sort of polarized political situation right now. I think as we move forward, we scientists have a responsibility to explain the benefits of sharing this data and making this available without any cost to the patient, either financial or in any other way. And I think you're right that I think that no one can guarantee that that would be true to the extent that laws change and policies change. However, I do think that the Broad has been at the forefront of policy advocacy as well, and we have been very vocal at every level of government with the idea of what is the right thing to do in this area. So to the extent that we as scientists can add our voices to that debate and sort of speak from perhaps a place of understanding of what the implications are, hopefully those voices will be heard and things will move forward in that direction. Well, thank you. Yes, sir, you've been very patient.
The drug that was used on Mary was developed for some other situation initially.
Let me try to. Thank you.
The drug that was developed for some other situation that was used on Mary, initially was it addressing certain markers, and those same markers appeared in Mary's situation?
Oh, I see what you mean. Yes, yes, I will repeat the question. The gentleman asked if there were certain markers in Mary's situation that were common to the rheumatoid arthritis, which is the other disease for which this treatment was developed. And the answer is yes. So work that was done in the Mundell laboratory, and Peter Mundell is sitting in the background there. The discovery was that there's a marker or a molecule called CD80, and that is the target of this drug, of abatacept. And it appears that people thought that it is relevant to T cells in the immune system, and that's how it is relevant to rheumatoid arthritis. But Peter's lab showed that this molecule is also important in podocytes, the cells that I told you about at the filter barrier of the kidney. And so with his fundamental work showing that this marker or molecule is relevant to the disease that we were talking about today, nephrotic syndrome, then I was able to take that drug and say, okay, there's a scientific rationale at the molecular level for giving it to Mary.
Yeah, that's what I was wondering.
Yeah. We are trying to expand that concept now because at first, and I will say I was perhaps naively thinking that we would be able to stain the kidney of Mary or other patients for that molecule, and that would be a marker that we can use to decide when to give this drug. We thought that was a great idea. I thought that was a great idea, and that did not pan out. I'm full of ideas that didn't work out, in my scientific career. But we are now working on a genomic approach to that. So we're trying to see if there's something about Mary's genomic information that can guide us as to why she's a responder, an exceptional responder, and why other patients do not respond. And we're sort of hot on the pursuit of something there, but we don't know yet if that's going to work out. So we'll see.
Let's thank Anna.
Thank you very much.